We're going to spend a lot of time on the axis growth, or the axon growth of neurons. This plays a key role in things like Huntington's disease and Parkinson's disease and a number of neurodegenerative disorders that we're looking to cure today, which is why I want to spend a lot of time on this over the next few days, because this is really what we need to gather from developmental biology, is the understanding of how not only to regenerate neurons, but how to regenerate them in the way that will repair and restore functionality that comes about from these problems that are so prevalent. We have no cure for Huntington's disease. We have no cure for Parkinson's disease. We have drugs that can slow the progression of things like Huntington's disease. And we have some types of stem cell therapies to aid in restoring some functionality in Parkinson's disease. But we're still a long ways off from curing or completely getting rid of these being a problem. So I don't think we'll be able to get, I will get into the dynamics of what causes Huntington's disease and what causes Parkinson's, as well as what this is related to in terms of neurogenesis and how the axons form and find their targets. The other main thing that we're going to go over is the dynamic process of how neurons find their targets, because this gets into spinal cord injury and therapies, where when you lose limb functionality because of a spinal injury, how do we reconnect it? How do we bridge the gap and bring them back to the right connectivity? We have to understand how axons grow and how they find their targets. During early development, it's a very complex process. So we're going to go through the dynamics of how that's done. It's even more difficult to do dealing with an adult, dealing with an already developed system where the axons, some of these axons are 2 to 3 feet long. So when you sever one, axons naturally only grow about an inch a year. Okay? They don't grow that fast. We can stimulate the artificial growth of them, but it's not about their growth. It's about their direction. It's about finding their targets and restoring that functionality. Imagine that you had a computer, all these computers, you know, with all the same colored wires, all white wires, and you sever that and be like, okay, put it back together. Make sure each connection is right. It's not impossible to do. However, there are techniques that are being done, especially in development, to actually label neurons to be able to see where they're at. This will show that if you look in a supplemental part of your book, it talks about how they're able to put this gene in various neurons that creates a fluorescence. It's called the Krelok's recombination. Anybody ever heard of Krelok's? It's basically a way of putting a substance into a cell that causes recombination of some of the genetics. Well, the long and the short of this is it allows to generate over about 100 different colors with this Krelok's recombination, which therefore allows us to follow individual neurons amongst this huge mess of neurons in the brain and in the spinal column, which will allow us to say, well, hey, where do these neurons, where are they? Let's see if we can rewire these things going on. The brain bow is one of the newest techniques in developmental biology to look at where these axons are going and actually getting a coloration of them. These are all of the factors of neurogenesis. You've got induction, you've got birth and migration. We talked about that in the neural tube and how when they're born, how far out they migrate, and therefore what signals are they associated with. They take on specific fates depending upon whether they're in the dorsal or the ventral neural tube. Now, this is where we're going to focus on. How are the axons, when they project out of the nuclei of the developing neurons, how are they guided to their specific targets? And this is not just an embryological question. It's also a developmental question. And this is where Huntington's and Parkinson's disease comes into play, is that due to the plasticity of neurons, there typically must be a continual signal of paracrine factors that allow the neurons to be maintained. And when those paracrine factors in your brain start going away, neurons start dying. And this is what causes Huntington's disease. And this is what causes Parkinson's disease. And this is the holy grail of figuring out what's going on, is by saying, well, how do we sustain these neurons? How do we make it so that these proteins are constantly being secreted to maintain the plasticity of the brain and so that neurons don't die? Okay? So once the synaptic connections are forming, we look at survival trophic factors. They're called neurotrophic factors, and they're constantly being produced in your brain to keep these connections between the neurons and their respective tissues and our other neurons. Okay? So, brief overview as far as motor neurons. These, as well as other types of synaptic neurons, the main body, or the soma of the cell, is where the nuclei is at, and then you have the axon hillock. The axon, again, can be really, really short, or it can be several feet long. It depends upon the neuron in which you're dealing with. Most neurons, especially in the brain, can actually receive connections from about 100,000 other neurons. So these dendrites that are projections from the main body of the cell can actually form other connections with other neurons. That's how complex our brain is. You have just trillions and trillions of these connections going on between all of that, which creates just such a complexity that is just, you know, astronomical in its proportions to try to understand what's going on. Okay? As neurons develop, and as muscles start to develop, and as limbs starting to extend, the axons have to find their targets very early on. And once they find their target, then they stay with that target as the limb grows and as the body starts forming its various tissues. So this is one of the reasons why it's so difficult to get axons in an adult to go back to their respective tissues, is because in development, they find that area that they're supposed to synapse with very early, and then as that area continues to develop, then it reforms that connection and extends out with it. So you don't have the limb forming and then the neurons trying to go all the way through the limb to find their target. They form and follow with the limb as it's forming. So they don't have a huge distance to go to find their initial target, and then they stay with that target all the way through development as the various parts of the embryo are forming. Neurogenesis. When a neuron forms, let's go over the anatomy of a neuron. I know some of you have already had this, but others have not. So the anatomy of a neuron, the main body of the neuron is typically where the cell nucleus is at. And this main body has projections coming out of it, which we call dendrites. Now, these dendrites can be so highly branched that you can form hundreds of thousands of connections with other neurons coming into that. So a single neuron can synapse with tens and hundreds of thousands of other neurons. This is why the complexity of the brain is so immense, is because you have billions of neurons and even more so connections between all of these that are forming these pathways of memory and cognition and things of that sort. Now, the main part where the functionality of a neuron comes in is in the axon. Now, the axon can be fairly short, or it can be up to a couple feet long, depending upon what type of axon you're dealing with. Some axons project from your spinal column and go all the way down your leg, down to your toes. Those are the ones that are a couple feet long. So the question becomes, how do both motor neurons, which affect your skeletal muscle tissue, as well as your sensory neurons, which typically receive sensory information from your peripheral nervous system as well as from all associated organs internally, and integrate them into your brain and your spinal column. And it's a very complex process. The holy grail of this is, when you get nerve damage, how do you remake those connections? When you get spinal cord injuries or brain injuries in which you try to reestablish these connections. And that's really where things become extremely difficult. You'll see why, in development, these have to happen fairly early on, even for them to be able to reach their targets and synapse, which means connect with, the various tissues that are necessary for their function. So you remember about the neural tube. What is some of the main factors that cause the patterning of the dorsal ventral axis of the neural tube? What were the two main groups of families of morphogen factors? Sonic hedgehog, okay? Which area of the neural tube expresses sonic hedgehog? The notochord, which is a chord of mesoderm, part of the mesoderm, will induce the ventral dorsal neural, or the ventral neural tube, which we call the floor plate, to start expressing sonic hedgehog. So as sonic hedgehog is being expressed in a morphogen gradient factor, this is going to turn on certain genes, especially in the ventral region, depending upon the threshold or how much sonic hedgehog is actually being expressed. What about in the dorsal region? Which gene or family is typically expressed there? BMPs, bone morphogenic proteins. These work as an antagonist to sonic hedgehog as well. And these form a gradient from, they are initially induced by the roof or the ectoderm, and then they induce cells which we call the roof plate, and these will then create a gradient along the dorsal ventral axis. These two gradients ultimately cause certain genes to be turned on at different levels all along the dorsal ventral axis. Especially here in the ventral axis is where we're going to pay particular attention to, because that's where motor neurons form in your spinal column. And the motor neurons which will cause your limbs and your muscles that are under voluntary control to be able to fire and do what they're supposed to do. Here's what we're looking at. Each neuron, now do you remember how the neuron cell bodies progress internally to the outward region? Which the cell bodies stay more in the ventricular zone of the neural tube? The early or the late-born ones? The early-born ones. So the earliest neurons that are born, the cell nuclei remain here and the axons project outwards. Well, as the other neurons are being born, they have to pass through the early-born neurons, and those receive inductive signals from those early-born neurons that will then help them in their differentiation. Some of these neurons, you can see here, it matters where they're at in this mantle zone. If they're a little bit closer, they're going to express certain genes. If they're a little bit further away, they're going to express other genes. And that's what predetermines where their targets are going to be. So there are three kind of main mechanisms that a axon has to go through to be able to find its target. The first neurons that project out are the motor neurons. And then the sensory neurons will project out along the same path as the motor neurons. And that's what we call pioneer nerve fibers. This ensures that the sensory neurons and the motor neurons affect the same tissue. Okay? So all you need is a couple pioneer nerve fibers to find their location, and then all the others can just follow that path that they find out to it. So let's talk about the